everyone and welcome. In this video we're checking out February-March 2024. Before I get started, make sure to check out the notes I have available for you in the description. There are notes for A-Level Bio, Chapter 1, Energy and Aerobic Respiration. Still more notes are coming and they're on the way, but for now these are the ones available and they're available for free. Also, if you have any comments, any questions, make sure to leave them down in the comments. Now let's go ahead and get started with question number 1. A student used a light microscope to observe a blood smear on a microscope slide. An eyepiece graticle was used to measure the diameter of a white blood cell on the slide, and the student recorded that the white blood cell was 5 eyepiece graticle units in diameter. What additional information does the student need to determine the diameter of a white blood cell in micrometers? Okay, to determine the diameter of a white blood cell, you have the eyepiece graticle units, but we need to know the length of the eyepiece graticle units themselves. To do that, we calibrate it using a stage micrometer, and so we need to calibrate the eyepiece graticle using stage micrometer, which is exactly A. B says calibration of eyepiece graticle using a stage micrometer and the magnification. Actually, magnification plays nothing into this, and so no, not magnification, and so 1 should be A. Let me change the color because I don't know why I'm using this. Let's use blue. Which statement explains why it's necessary to use an electron microscope to see the cristae of a mitochondrion? Okay. Why it's necessary to use an electron microscope? Now, the light microscope is not going to allow you to see the cristae. The cristae are the inner membranes, the folds of the mitochondria that you, see in pictures. Now, these are 7 nanometers in width. Okay, the light microscope here does not have an incredible resolution. Okay, its resolution reaches only to 200 nanometers. So anything closer than 200 nanometers will be really blurry and we won't be able to see it. Now imagine, for example, you have a camera, right? The camera, the higher the resolution, the better the quality of the image. If the resolution was lower, then anything, and let's say the two buildings that you can see, for example, in real life will be merged into one in the background. We won't be able to see them because the resolution is really low. And so anything closer than a certain distance will not be seen. Okay, it's not about magnification, it's about resolution. And so here, let's see. The magnification of the electron microscope is greater than the light microscope. It's not about that. The magnification of the light microscope is pretty high. It's not about the magnification, it's about resolution. It's a limiting factor for magnification. The membranes of cristae are separated by a distance greater than 200 nanometers. Actually, it's less. It's 7 nanometers. The reason we cannot see it. The maximum resolution of a microscope using light is very low. Exactly. So that's C. Some stains can be used to identify cell structures in living cells. A dilute solution of one stain causes the whole cell to appear blue. This blue color rapidly disappears from most cell structures, and no cell structures that release energy actually stay blue. Interesting. Which type of cell structure is likely to be blue? It's pretty easy. It's the mitochondrion, because the mitochondrion is the site of aerobic respiration, and that is the site of release of energy. Okay, 4. When mucus is secreted from a goblet cell, these events take place. We have addition of carbohydrates or protein, fusion of vesicle with the cell surface membrane, extracellular release of glycoprotein, separation of vesicle from the Golgi body. So what we have here is mucus is supposed to be a glycoprotein. Now, where glycoproteins are formed, it is in the Golgi body. Okay, so first we're going to have a protein formed at the ribosomes, at the rough endoplasmic reticulum. The proteins are going to then move in a vesicle all the way to the Golgi apparatus. They're going to fuse with the Golgi apparatus, where they're going to be modified, and then they're going to be added to, let's say, a carbohydrate forming a glycoprotein. The glycoprotein is going to be now that this is the vesicle that came in. This is the Golgi body, and now the glycoprotein is going to escape in a Golgi vesicle, which is going to move across the cytoplasm guided by the microtubules of the cytoskeleton and fuse with the cell surface membrane. And so 1 should be the addition of carbohydrate to protein. That's 1. Okay. 2 should be the separation of the separation of the vesicle from the Golgi body. 3 is going to be the fusion, fusion with the cell surface membrane, and so that should be 2. And lastly, we have extracellular release of a glycoprotein. 1, 4, 2, 3, which is A. Okay, the diagram shows 4 biological molecules. You have DNA, RNA, capsid, and 70S ribosomes. Which features are present in typical prokaryotes? DNA, RNA, and 70S ribosomes. Capsid is a structure typically found in viruses. The capsid is supposed to be the protein code formed of little protein units called capsomeres. Okay, so that's not going to be found in prokaryotes. That's 1, 4, and 3, which is C. 6. Which rows shows features that occur in dicotyledonous plant cells and also in bacterial plant cells? Interesting. So, what's it called? Let's see. 70S ribosomes, 80S ribosomes, centrioles, and circular DNA. Okay, we both have circular DNA. Obviously, with we, I mean plant cells and typical bacterial cells. Do they have centrioles? Actually, neither the plant cell nor the bacterial cell have centrioles. Centrioles are only found in animal cells. Okay, so this is an X. 80S ribosomes are not found in typical bacteria. Okay, they only have 70S ribosomes. 70S ribosomes are found in both, so that's B. Which flowchart outlining the test for non-reducing sugars is correct? Okay, non-reducing sugars, like let's say sucrose. Firstly, you're going to have to firstly you're going to have to break down the, what's it called, you're going to have to hydrolyze, okay, break down, okay, hydrolyze the non-reducing sugar, because there is no test for non-reducing sugar. We have, however, tests for reducing sugars. So we're going to have to break down the non-reducing sugar and see if it tests then the solution after the breakdown. See if that solution tests positive for reducing sugars. Because let's say, what's an example of non-reducing sugar? That's sucrose. Sucrose is a disaccharide. Disaccharides, when broken down, turn into monosaccharides. All monosaccharides that you know are reducing sugars. And so if there was a non-reducing sugar, once it's broken down, the test should be positive. And so we have, let's see the options. Boil a sample, add alkaline sodium hydrogen carbonate to sample, neutralize with dilute hydrochloric acid, add benzoic. Okay, that does not make sense. Why are you adding a base in the beginning? We hydrolyze in the presence of an acid. Okay, so this is wrong. Add alkaline sodium hydrogen carbonate, again, wrong. Boil, sample with dilute hydrochloric acid, okay. Then you neutralize the hydrochloric acid so it does not affect the Benedict solution, yes. Add Benedict solution and boil, let's see. Okay, which diagrams show the release of a water molecule during the formation of a glycosidic bond? Okay, here you could see we have a formation of a glycosidic bond. Here you could see we have a formation of a glycosidic bond. Here you could see we have a formation of a glycosidic bond, 1, 2, and 3. That's A. Okay, which molecules have a structural formula that contains C-double-O bonds? Let's see, amino acids, sure they do. How do amino acids look, guys? Okay, amino acid is supposed to be an alpha carbon. We're supposed to have nitrogen and then 2 hydrogens, and then here we have an R group, right? And then here we're supposed to have an NH, and then we have a double bond O, and obviously OH. Okay, so that's an amino acid. What do fatty acids look like? Well, fatty acids are supposed to be double OOH and then a fatty acid chain, right? And then we have glycerol. Glycerol is an alcohol. Alcohols do not have double O, C-double-O bonds. Alcohols look something like this. Pretty boring. A bunch of OH groups and, really boring. Anyway, what's it called? So, yeah, obviously glycerol is not one of our options. Protein. If proteins are made of amino acids, then proteins sure have C-double-O bonds. So it's 1, 3, and 4. That's C. Which statements about triglycerides is correct? In any triglyceride, all fatty acids are saturated or all fatty acids are unsaturated. Actually, no. Sometimes one of the fatty acid chains is going to be saturated, and then another one will be unsaturated, and then the other one will be saturated. So no, this statement is totally false. Okay. 2 triglycerides are joined together by an ester bond. Okay, actually, no. It's the fatty acid and the glycerol that are joined together by the ester bond, not 2 triglycerides. So that's wrong. Triglycerides are polar hydrophobic molecules. This does not make any sense. So okay. Glycerol is joined to each fatty acid by a covalent bond. Yes, actually, ester bonds are covalent bonds. Phosphodiester bonds are covalent bonds. Glycosidic bonds are covalent bonds. All these molecules joining together, they are molecules. And molecules joining together, they have covalent bonds, not ionic bonds. These are ions. These are elements, right? Compounds joining together, not molecules. I think you can see the difference. I'm not really a chemist, but I hope that was clear to you, so that should be D. Okay, 11. Many flowers produce a sweet solution called nectar. Bees provided with nectar enzyme Q to change the nectar into honey. Interesting. So after testing a sample of nectar in the presence of reducing sugar using standard laboratory reagents, the sample was blue. After testing the sample of honey in the same way, the sample was orange. So we're testing for reducing sugars. So we understand that nectar is not a reducing sugar, but however honey has reducing sugars, and so which conclusion about reaction catalyzed by enzyme Q is consistent with these results? Now, as we told you guys before, Benedict's solution is supposed to be a solution testing for reducing sugars. And so if you, let's say, have a non-reducing sugar, once you break it down, once you hydrolyze it into a reducing sugar, the test should end up being positive, and that's a test for non-reducing sugars. And so that means that the nectar here is supposed to be non-reducing. There was hydrolysis that happened and that produced a reducing sugar, and that's it. Okay, so let's see. Type of reaction, it's hydrolysis. Substrate, it's sucrose or maltose, obviously sucrose, because the substrate is supposed to be a non-reducing sugar. Sucrose is the only non-reducing sugar that we know so far. Product is supposed to be fructose. Fructose is a reducing sugar. All monosaccharides are reducing sugars. So that's C. Okay, 12. The diagram shows the structure of part of a peptidoglycan molecule. Okay, so we have peptidoglycan, and here it says which type of 1 to 4 bond and how many peptide bonds are shown in this part of molecule. Okay, so let's see. What is the 1 to 4 linkage that we have here? Is it alpha? Is it beta? Now, since you see that each one of these is in different orientations, so we have the same molecule but different orientations, so I would suggest that this is obviously a beta. 1,4 bond. Okay, however, number of peptide bonds that we see, let's count. Okay, so what does a peptide bond look like? You should memorize this. It's a C-double-O bond with an H, okay, and then whatever connections we have here. Okay, count them. First peptide bond I see is this one. Second one is here. I mean, yes, they don't show the double bond, but it's still a CO with carbon and oxygen. It's always double bond, okay. And then here again and here again, so we have 4 peptide bonds, and so that is beta 1, 4, and 4, so that's D. A scientist investigated the progress of 2 enzyme catalyzed reactions in separate test tubes, X and Y. Both reactions result in color change that can be detected using colorimetry. 0.5 centimeters cubed samples were taken from each test tube at the start of the investigation at regular intervals for the next 5 minutes. Copper ions were added to each sample as soon as the sample was collected to inactivate the enzymes and stop the reaction from progressing any further. Okay, so the absorbance of each sample was measured using the colorimeter. The graph shows the results of this investigation. So we have the investigation showing the percentage absorbance over time, and then that's it. Which statement is consistent with the results shown in the graph? We have X and we have Y. Substrate in X has higher absorbance than the product. Okay, so let's see. Substrate in X. So obviously here we start with the substrate, we end with the product. When we are no longer doing the reaction, we're supposed to be finished, and so that's the product. And so we could see here that the substrate, percentage absorbance is pretty high, but not higher than the final product, obviously. And so no, I would say substrate has lower absorbance than the product. The product in test tube Y, however, has lower absorbance than the substrate. Actually, true. The product here, you could see it's at lower percentage absorbance than at the start with the substrate, and so I would say B is correct. Let's just check out the other solutions. The rate of reaction in test tube X increases with time. The rate of reaction will increase with time. I would suggest that it doesn't increase with time, because you could see how we start off with a constant rate of reaction, then the rate will decrease as the line becomes, the change in percentage absorbance over time is just decreasing. And let's see, so this is also wrong. The rate of reaction in test tube Y increased with time. I think same thing happened for test tube Y. It's just that the reaction produces a product with different colors, and so instead of having percentage absorbance being high, it's low, and that's just fine. It does not mean that we are faster or slower. It just means that the product here is let's say lighter in color. Okay, so D should also be wrong, and so B is my answer. Okay, 14. A student investigated the hydrolysis of lipid in high-fat milk using the enzyme lipase. Okay, so we have 1 centimeters cubed of enzyme solution was added to 10 centimeters cubed of high-fat milk. The temperature was kept constant, and the pH of the reaction mixture was recorded at time 0 and every minute for 20 minutes. Interesting. Which statements correctly explain describe the expected results of this investigation? So let's see. The product forms more slowly as time proceeds because the concentration of substrate is decreasing. That is true. that does happen after a while because we're not really like adding excess substrate and adding what's it called substrate as we go on. No, actually substrate gets used up, and so the rate of reaction should decrease. And so this is correct. The pH of the reaction mixture increases rapidly in the first few minutes, and then in increases less rapidly. Actually, with this type of reaction where we have the hydrolysis of milk, actually hydrolysis of lipid in milk. Lipid is supposed to be broken down into fatty acids and glycerol. Fatty acids are an acid, and acids decrease pH of surroundings, and therefore pH should decrease, not increase, and so it's wrong. The increase in concentration of product will eventually cause the lipase molecules to denature. Yes, actually that's just because of the same exact reason we mentioned earlier. pH is going to decrease because of the fatty acids, and pH will affect the tertiary structure of the enzyme, which will result in its denature. And so that is true. So yes, 1 and 3, that's C. Okay, what is the correct range of measurements for the width of the cell surface membrane? The cell surface membrane is stated as 7 nanometers in width, and that's 5 to 10 nanometers should be your range. So that's B. 16. Where in the cell surface membrane are the carbohydrate chains of glycoproteins and glycolipids mainly located? Okay, glycoproteins and glycolipids, where are they mainly located? They're mainly located on the outer surface because they mainly act as receptors, as adherence molecules. They help cells to attach to each other. They help, they act as antigens. So they're mainly located on the outer surface. Okay, sodium ions can enter cells across the cell surface membrane. Which methods can be used by sodium ions to cross the cell surface membrane and enter a cell? Okay, so is it active transport only, or is it active transport, facilitated diffusion, or is it facilitated diffusion and simple diffusion, or is it simple diffusion only? I would suggest it's facilitated diffusion and active transport. Obviously not simple diffusion because they are ions and they're polar and they can't really cross the cell surface membrane with hydrophobic inner core, and so obviously that should be B. Using active transport or facilitated diffusion, both will use protein channels or protein carriers, and so they're able to cross that pretty fine. And so 17 is B. Okay. The diagram shows an artificial partially permeable membrane was used to separate a 5% sodium chloride solution and a 10% sodium chloride solution in a beaker. Those two sides of the beaker were labeled R and S, and you could see here we have a less concentrated R and a highly concentrated S. Okay, so which row correctly explains what will happen in the half of the beaker labeled S? I can see here that we have a partial membrane. I think this will be a question about osmosis, because osmosis definition is supposed to be movement of water from region of higher water potential to lower water potential across a partially permeable membrane, and we have that just here. And so description of S. Volume of solution increases. Okay, let's think together what exactly is going to happen. Obviously there will be water movement because, as mentioned, this is an osmosis question. It's partially membrane for a reason. And where will water move from? Okay, so let's say that you have a cup filled with sugar. Okay, the more sugar you put, the less water there will be inside that cup, right? So if we have 10% sugar, that means we have more sugar and therefore less water. And so in this case, obviously this is like salt, not sugar, but whatever. Okay, let's just, whatever. Okay, go with the flow. So this means that we have less water here than on the other side. So this is less water, okay, lower water potential. And then on the other side, we have 5% sodium chloride, and that means that we have a higher water potential, more water on this side. And so how does water move, guys? Water will move down the water potential from where we have more water to less water until we reach equilibrium, until we have the same amount of water on the left side, on side R, and the side S. And so the movement will be like this. And so the volume here is actually going to increase. I mean, obviously this is not how it's going to look, but yes, the volume of water is going to increase. Volume of solution S is going to increase, and the explanation is because movement of water from higher water potential to lower water potential, and so that is A. Okay, 19. Agar cubes can be used to demonstrate the effect of using on diffusion of changing the surface area to volume ratio. And so 3 different agar cubes were made using a colored indicator solution were passed into dilute acid that diffused into these cubes. As the acid diffused into the agar cubes, the color of the indicator solution actually changed. So the cubes had volumes of 1, 2, and 3 centimeters cubed respectively and were left to dilute for 10 minutes. Okay. And so all other variables were kept the same. After 10 minutes, the agar cubes were removed and the dilute acid was cut in half, and the cut surfaces were observed and the results were recorded on the diagrams. All diagrams are drawn to the same scale. The results for the 2 centimeters cube are shown here. You could see that part is what's it called of the acid diffused into the agar cubes and not changed its color. And so which diagram shows the results for 1 centimeters cubed and 3 centimeters cubed cubes? Okay, interesting. So let's figure out the surface area to volume ratio of this cube. Okay, so the volume of the cube is supposed to be the side times side times side, and so that's side squared. To find the side cubed, to find the side, you just take the cubic root of 2, and that's the length of this side. Okay, and so the surface area to volume ratio here should be, well, we know surface area is supposed to be the area of this surface, is supposed to be the area side times side. And so if you multiply this cubic root of 2 by cubic root of 2, you will get an area of 1.587. Now if you get this and you divide it by 2, then you get the surface area to volume ratio of 0.793 or 0.794. Okay, now this is supposed to be surface area to volume ratio for cube 2. Now let's try it for other cubes. Other cubes here are going to have a volume, this one for example, of 3, and therefore each side will be cubic root of 3 units long, or 1.4422. Okay, now if you are going to multiply this number, cubic root of 3 by cubic root of 3, you get the surface area of the cube. So this is the surface area of cube 3 is going to be 2.08. If you were now to divide it by 3, you should get 0.69. And so you could see that the surface area to volume ratio of the last cube is actually smaller. And then for the first cube, the last, one, we'll do the same procedure. We'll take the cubic root of 1, right, and that should be 1, and we'll multiply 1 by 1, that should be 1, we'll divide 1 by 1, and we'll get 1. How surprising. And so 1 is the surface area to volume ratio of cube, the first cube, and so obviously it has the greatest surface area to volume ratio, and so there should be lots of diffusion here into cube 1, but there should be less diffusion into cube 3. Now here, if we look at the options that we have here, they were left for 10 minutes and only 10 minutes. Obviously, we won't have this situation because the surface area to volume ratio for cube 1 is far higher, and so we should have more diffusion in cube 1 than in cube 3, and obviously we won't have this situation over here where in 10 minutes the cube just goes magically white. Actually, no, it's also not going to be A, because again, we only have 10 minutes and for the acid to completely dissolve and change the color, it's quite impossible. So I would say it's going to be C. Okay. 20. During sperm formation in mammals, part of the structure of each chromosome is replaced with proteins called protamines. This replacement allows DNA to be packaged more densely than would otherwise be possible. Which part of the chromosome is replaced by protamines? Okay. Which part is replaced by protamines? So this allows DNA to be packaged more densely. What allows DNA packaging? That's the histone protein. Okay, so we have chromosomes, and chromosomes are made of DNA and histone, and DNA will wrap around histone. Alright, so if you want more packaging, more wrapping around histone, then you'll need to replace the histone by the protamines, and so C should be your answer. The diagram shows the mitotic cell cycle, during which phase do chromosomes condense and become visible. Okay, where do chromosomes condense and become visible? This is cytokinesis. Okay, so this is cytokinesis, this is mitosis. And if this is mitosis, this is the G2 phase, this will be the S phase, the synthesis phase, and this will be the first growth period, G1. And so when do chromosomes condense? That's in prophase. That's the first phase of mitosis. Okay, so that's going to be D, in mitosis. Okay, which statements correctly describe features of stem cells that are essential for their role in cell replacement and tissue repair? After mitosis of stem cells, the daughter cells can remain as stem cells or follow a developmental pathway that leads to the formation of specialized cells. Sure, that is effective for their role in tissue repair, because in tissue repair we need to replace cells. Okay, so we need to differentiate into these types of cells that we need to replace. And so that's 1, that's correct. 2, the stem cells are different to all body cells because they retain all the genetic information in their DNA throughout the life of the organism. Actually, all what's it called cells retain their DNA. It's not just stem cells that retain all its genetic information. So no, a small population of cells is stem cells retained in the body of adults throughout their lifetime. Actually, yes, we do not have many stem cells. They do not have the maximum power. They are called, what's it called? Multipotent stem cells that have, like, power to differentiate into cells according to where they are. So let's say cells in the bone marrow are able to differentiate into blood cells. However, what's it called? Yes, we do have a small population, and I think that is part of stem cells. That is a true feature about stem cells. Okay, my pen died. Stem cells have more telomeres than the other body cells, and this allows them to undergo an unlimited number of mitotic divisions. Actually, stem cells have the same number of telomeres as all other body cells. It's just that theirs are always longer because they have the telomerase enzyme. And so 4 is wrong and 3 is right, so it's 1 and 3 only. 23. Which statements are correct for all nucleotides? The nitrogen-containing base is always attached to carbon atom 1 of pentose. That is true about the structure. Phosphate group is always attached to carbon 5 of the pentose. That is also correct. A condensation reaction occurs to join nitrogen-containing base to pentose. I mean, how other way will the base join to the pentose sugar? I think it's also true. Nucleotides are linked together by condensation reactions between phosphate groups. No, actually, the condensation reaction will happen between one phosphate group and one hydroxyl group, and the bond is called the phosphodiester bond. Okay, so what's it called? This is wrong. 4 is wrong. So it's 1 and 2 and 3, and that's A. How many phosphodiester bonds are present in a circular DNA molecule of 2,700 base pairs? Okay, let's think about this. 2,700 base pairs means 2 strands, each one with 2,700 nucleotides, and so 2,700 times 2 should be 5,400 bonds. That's how many. Okay, so that's D. Okay, which statements about complementary base pairing are correct? Purines and pyrimidines are different sizes. Complementary base pairing occurs during translation. The base pairs are of different lengths. Uracil forms 2 hydrogen bonds with adenine. Sure thing, that does happen. The base pairs are of different lengths. Actually, no. A base pair is formed between a purine and a pyrimidine. A purine being a nitrogenous nitrogenous base that has double ring structure. A pyrimidine is a nitrogenous base with a single ring structure. And so if it's always between purine and pyrimidine, then it should be double ring, single ring. That's 3 rings. That's the same size always. And so it could be between adenine and thymine, and that's the same size as between guanine and cytosine, because guanine and adenine are purines, and cytosine and thymine are pyrimidines, and so that's wrong. Okay, 2. Complementary base pairing occurs during translation. Does it happen during translation? Sure it does. Purines and pyrimidines are different sizes. That's true. Purines, double ring structure, pyrimidines, single ring. And so it's 1, 2, and 4. That is B. During the mitotic cell cycle, the chromosomal DNA is replicated. The specific points in DNA molecule where replication is occurring are known as replication forks. A typical human chromosome has about 150 million base pairs of DNA. It takes about 1 hour to replicate the DNA of a typical human chromosome. The rate of replication using a single replication fork is approximately 50 base pairs per second. Approximately how many replication forks must occur in a typical human chromosome during DNA replication? Interesting. So one replication fork should have 50 base pairs per second, right? So we have 50 per 1 second. Now instead we have 150 million base pairs, and that's per 1 hour. Okay, so 150 million and then per 1 hour. Now let's convert 1 to 60 times 60, because in 1 hour we have 60 minutes, in 60 minutes we have 60 seconds each minute. Okay, now this rate, is supposed to be the rate of formation of the replication forks in DNA. We want to know how many replication forks are formed, and so if we multiply the rate of formation of one replication fork, then we multiply this by x, and we figure out how many replication forks are formed. So again, x is the number of replication forks, this is the rate of formation of one replication fork, and this is the formation of replication forks in DNA. Okay, so if we get the number on the other side, 150 divided by 60 times 60, you will get 1 over 24 equals to 50x. If you divide by 50 on either side, you will get x equals to 1 over 24 divided by 50, which will be 8.33. Now this, ladies and gentlemen, is wrong because we forgot this is a million situation, and so you multiply here by a million. Okay, So you multiply what we got by 1 million, and so that should be 833, which is approximately 835, which is A. Okay, which molecule has its synthesis directly controlled by DNA? Okay, so synthesis directly controlled by DNA is amylase, cholesterol, glycogen, or phospholipid. It's definitely amylase because amylase is the only protein here. Protein synthesis directly controlled by DNA, because transcription is basically we're like taking DNA and we're making our mRNA out of it. So like A. Okay. Which statements correctly describe the association between a companion cell and a sieve tube cell? Okay, so the companion cell provides all the ATP used for energy requiring processes in both types of cells. Actually, no, the companion cell, this is not the role of the companion cell. The companion cell will provide a nucleus for both cells. Okay, so I would say then what's called companion cell provides controls the active transport of molecules through the sieve tube plates. There is no active transport through the sieve tube plates, and the companion cell provides side-to-side movement of assimilates between sieve tube cells. Between sieve tube cells? It does provide side-to-side movement between the companion cell and the sieve tube cell, but not. There is no side-to-side movement between the sieve tube cells. This does not make sense to me. Okay, so C is wrong, B is wrong, and A is wrong. The companion cell provides a nucleus that controls cellular activities in both types of cells. This works, and so D is my answer. 29. A maize seedling was grown in soil that contained lanthanum ions and labeled with chemical that fluoresces under violet light. And so the diagram represents what was observed on a section of fruit was examined using a light microscope with ultraviolet illumination. We have fluorescence, we have root hair, we have Casparian strips, and we have xylem. Okay, so you could see here we had fluorescence, which means we had ions all the way around the root hair until we reached the Casparian strip. And the Casparian strip is known to block the Laplace pathway because the Casparian strip is a waterproof material. It was a substance that's it's basically this substance that's made out of suberin, which is a waterproof material that will be placed in the cell wall and therefore stop substances from moving in the cell wall, and the cell wall forms like a whole lot of parts of like the Laplace pathway, the dead pathway, because look, water and mineral ions can flow either through cells or they can flow through the freely permeable cell wall and empty intercellular spaces. And so there were one of two options, and if the Laplace pathway being the cell wall and the intercellular spaces, if it's blocked, then that means that we're flowing through the Laplace pathway. Okay, but in this case you could see here that there are no ions anywhere else, and so obviously when the Laplace pathway was blocked, that means that the ions couldn't move anywhere, and so the ions are not able to move in the Laplace pathway. And so the ions are not transported through the Laplace pathway or the Laplace pathway. No, they're transported through the Laplace pathway, however, not the Laplace pathway, that's B. Okay, so again, let's repeat what happens here. So we have ions that could be that could move, through the cell wall, through the intercellular spaces just fine. And these pathways, this pathway through the cell wall, intercellular spaces is called the apoplast pathway. And it's the pathway of least resistance because we do not have the partially permeable membrane, we do not have the organelles, we do not have the cytoplasm, the jelly-like material flowing, freely. And then some things cannot pass, once this barrier of the once this barrier surface membrane is present, the ions cannot flow through that. Okay, ions cannot handle it, and so they stop flowing. And so since they stopped flowing and they couldn't flow through the membranes, that means that they cannot flow through the Laplace pathway. And so B is the answer. Which description of adhesion and cohesion is correct? Adhesion refers to the force between water molecules due to hydrogen bonding. Cohesion refers to the force between water molecules and xylem vessels. That's totally the opposite. Cohesion is the force between the water molecules. Adhesion is the force that binds the water molecules to the walls. Okay, so adhesion refers to the reduced friction between hydrophobic lignin walls and the water molecules. I don't think so. There's no such thing as reduced friction. Okay, adhesion refers to reduced friction. Again, no. Adhesion refers to the force between the water molecules. And the xylem vessel walls. And cohesion refers to the force between water molecules due to hydrogen bonding. D is my answer. Okay, which conditions are needed to allow the mass flow of sucrose in phloem sieve tubes? Phloem sieve tube and sewer sources. Okay. And then So let's see. Higher hydrostatic pressure and higher water potential. Okay, so let's see. We need mass flow to occur in phloem sieve tubes. So what we need in sources is obviously a high, hydrostatic pressure, but actually a lower water potential. Okay, so that's called, sieve tubes. I mean, since we have sucrose and all this material, then this substance is going to flow, but we're not going to have such a high water potential. Then when we reach the sink, we need a higher water potential in order to continue moving. So there will be a high water potential there, and also a lower hydrostatic pressure, and so that should be B. Okay, now the hydrostatic pressure here allows for the mass flow in when we are in the stem, but then once we reach the sink, we need lower water potential. We need a higher water potential, which then can allow us to move into the cells of the root. Okay, one type of congenital heart defect is where the left and right atria are not completely separated. This is called atrioseptal defect. Interesting. So ASD is when the atria are not completely separated. ASD usually results in blood moving from the left atrium specifically to the right atrium. This causes the increased blood pressure in the right atrium and decreased blood pressure in the left atrium, which makes sense. Now, which row describes other effects caused by ASD? So let's just draw together the picture of the heart. Okay, so we have the left side of the heart and we have the right side of the heart. The right side being the deoxygenated side. Let's start with this. Okay, so we have the vena cava coming in here, and with deoxygenated blood we have the separation between the atria and the ventricle. Obviously it's not a box, you do not have a box inside you, but it just simplifies things. Okay, we have the ventricles, and then we have the ventricle being attached to pulmonary artery, and that is the right side. So we have the right atrium, the right ventricle, the pulmonary artery, the vena cava, and this is your left side. I'm sorry, the right side. Now for the left side, okay, what we have is firstly, okay, this is wrong because I cannot draw an opening now. Okay, so we have here the aorta, we have here the pulmonary vein coming in, and then we have the opening for the atrioventricular valves, and this is the left ventricle. So we have the left ventricle, the left atrium, we have the pulmonary vein, and the aorta. Okay, so how exactly does ASD work? We have the flow of blood from the left atrium to the right atrium, and so this increases the pressure in the right atrium, decreases the pressure in the left atrium. What happens to blood pressure in the pulmonary artery? Okay, in the pulmonary artery, okay, let me just make the color slightly darker because it's a little too bright. Okay, so what happens to the pulmonary artery? So since we have higher pressure here, we have higher pressure in the right ventricle, we have higher pressure in the pulmonary artery, and so it's increased. Blood pressure in the aorta, we have lower pressure in the left atrium, lower pressure in the left ventricle, lower pressure in the aorta, and so that is decreased. Okay, we have oxygenation of blood in the pulmonary artery. Okay, so since we have here the oxygenated blood, and here there's no oxygenated blood, and so now when oxygen flows from here to here, then obviously the oxygenation in the right side is going to increase, and therefore the oxygenation in the pulmonary artery is going to increase, and so it should be increased. And so yeah, it's increased, decreased, increased, and that's D. Okay, which property of water related to its role in blood and tissue fluid is correctly described? Okay, which property of water related to its role in blood and tissue fluid is correctly described here? Okay, so water is a solvent for all biological molecules. Water is a solvent for non-polar molecules. Water requires little energy to increase its temperature because it has high specific heat capacity. Water cools down slowly because it has high specific heat capacity. Okay, first option A, water being a solvent for all biological molecules, that works in the cytoplasm, okay, in a cell, but it does not really work in the blood. Okay, but this is not the function of water. Water is not there to act as a solvent. Water is there to it's just part of the blood plasma. Water is a solvent for most non-polar molecules. Actually no, it's a solvent for polar molecules. Water requires little energy to increase its temperature because it has high specific heat capacity. Actually, if it requires little energy, then it has low specific heat capacity. So the entire statement is wrong, and so C is wrong. This leaves us with D. Water cools down slowly because it has high specific heat capacity, and that makes more sense. Okay, the function of water in the blood being part of plasma, to dissolve some stuff, to carry some stuff, to be able to the fact that it has a high specific heat capacity, as that means that it will not increase suddenly and it will not decrease suddenly. And so that is D, part of homeostasis, part of, what's it called, the coordination of temperature. Okay, which statement about tissue fluid formation is correct? The hydrostatic pressure of blood decreases from the arterial end to the venular end. Actually, the opposite. Okay, so if we have what's it called, an arteriole, and then we have, okay, so we have arteriole, we have venule, and we have the capillary in the middle. Okay, now I don't know why the colors are like this. Oh, I think it's because of this. Okay, so here we have a higher pressure in the arterial end. We have high pressure, we have oxygenation, we have glucose, we have all these materials. Now these materials will go outside. Now when they go outside, obviously they're going to go outside, then they're going to move, and so obviously this changes the pressure. When everything leaves, all of a sudden the pressure here is going to decrease. The pressure inside the capillary will decrease at the venular end, and so stuff will move back, and so there will be drainage of the tissue fluid back. And so what happens here is the hydrostatic pressure of blood decreases from the arterial end to the venular end. It's the opposite actually. The water potential is always higher in the blood in the capillaries than in tissue fluid surrounding tissues. Actually, no, it changes. As I mentioned, stuff moves in and moves back, and so this changes the water potential. Okay, I don't know what happened to the pen. I think it's finished in charge. But anyway, B is wrong. C, no cells move out of the blood in the capillaries into tissue fluid. No cells move out of the blood and capillaries into tissue fluid. Actually, there are some cells. There are lymphocytes, the white blood cells, the phagocytes. These are going to be able to move. Most tissue fluid formation occurs at the venular end of the capillary. Most tissue fluid formation, actually no. The pressure. Oh, actually, A is right. I read it opposite. I thought that it said the opposite. As blood pressure will decrease from the arterial end to the venular end. That is true. You could see here pressure is higher and it goes down. So yes, A is right. But D is wrong. D is the opposite. Most tissue fluid formation occurs at the venular end. No, most tissue fluid drainage occurs at the venular end. Formation occurs at the arterial end, and that is because of letter A. The pressure at the arterial end is higher than the pressure at the venular end. 35. Oxyhemoglobin, carbamine hemoglobin, hemoglobinic acid, and carbonic anhydride are found inside our blood cells. How many of these substances will show an overall decrease in the concentration as the red blood cell passes through capillaries in the lungs? Okay, so in the lungs specifically, where at the lungs we have high concentration of oxygen, so high partial pressure of oxygen. So what exactly does that do? When we have lots and lots of oxygen, that obviously means we'll be having more oxyhemoglobin. Now, and at the muscle side, we had more carbon dioxide and we had more hydrogen ions because carbon dioxide was going to react with water, and that formed carbonic acid, and then carbonic acid will dissociate because it's a weak acid forming hydrogen ions. And these hydrogen ions will be in high concentration there. So again, at the muscle. So at the muscle side, we have lots of respiration. This respiration is going to produce lots of carbon dioxide. It's a waste gas. So carbon dioxide is released here. High concentration of carbon dioxide. What happens next is that carbon dioxide will react with water inside their blood cell. So we have plus H2O. This forms carbonic acid, H2CO3, in the presence obviously of an enzyme called carbonic anhydrase. Right, this catalyzes the reaction. Now, this is a reversible reaction, guys, so it happens in both directions. Now, H2CO3 is a weak acid. It breaks down immediately, forming hydrogen ions and HCO3 minus ions, okay? Hydrogen ions are going to be in high concentration at the muscle side, and they will react with hemoglobin to form hemoglobinic acid. And so hemoglobinic acid will be in high concentration at the muscle side, but obviously with oxyhemoglobin forming, hemoglobin is going to leave the hydrogen ions and ignore it totally for the oxygen, and that's why the concentration in the lung side of hemoglobinic acid will decrease. And so for the same thing for carbamine hemoglobin. Carbon dioxide can either undergo all of this, or it can directly react with hemoglobin to form carbamine hemoglobin. So obviously this will not happen in the lung side, so this will also decrease in concentration. However, carbonic anhydrase, which is an enzyme that is always present in our blood cells, it's not going to decrease or change in concentration. So which things will show an overall decrease? We have only two, carbamine hemoglobin, hemoglobinic acid. So that's B. Okay, which one of these structures typically contain cartilage and cilia? Okay, cartilage and cilia. Trachea, bronchioles, and bronchi. Bronchioles do not contain cartilage. Trachea contains cartilage, and bronchi do contain cartilage and cilia. So that's 1 and 3 only. Let's see. What statements about the function of tissues found in human gas exchange system are correct? Okay, so in the human gas exchange system, collagen in the bronchi prevents them from collapsing. It's not really the function of collagen in bronchi, it's the function of collagen in trachea. So we could cancel this. Smooth muscle in the bronchioles can contract to increase flow of air in the alveoli. Actually, if they contract, it means that they make the opening narrower, and so this does not increase the airflow. What's it called? Elastic fibers in alveoli stretch and recoil during breathing. That is true. What's it called? That does happen. So it's D, just 3 only. Antibiotic resistant strains of Mycobacterium tuberculosis are a major problem in treating TB. A new antibiotic, Tioxobactin, could be very effective at killing M. tuberculosis. With only a small risk that the bacteria will evolve Tioxobactin. Okay. Resistance. Penicillin and similar antibiotics bind to a single protein, but Tioxobactin binds to 2 lipids, interesting, that are needed for formation of bacterial cell wall. Tioxobactin binds to regions of 2 lipids that do not vary across many different species of bacteria. Which statements help to explain why that the use of Tioxobactin is thought to be less likely to lead to the evolution of antibiotic resistance than the use of many other antibiotics such as penicillin. Interesting. Okay, a single mutation can result in the bacteria and bacteria that are resistant to penicillin and similar antibiotics, but at least 2 mutations are required to produce Tioxobactin resistant bacteria. True. 100%. We need 2 mutations in order to produce Tioxobactin resistance. But 2 says mutations can affect the structure of proteins but cannot affect the structure of lipids because only proteins are made of amino acids. Okay, I understand that DNA does not directly affect the formation of lipids, but it sure has an effect on the formation of lipids. And so if there's a mutation, that should affect the formation of lipids. And so we have 2 is wrong. The lack of variation across many species of bacteria and 2 lipids that bind to Tioxobactin suggests that the particular cell structure of these lipids is essential for successful bacterial cell wall formation. I would say that is true as well. So it's 1 and 3 only. That's C. Okay. Monoclonal antibodies are now being used to treat some human diseases. What explains why monoclonal antibodies are suitable for this purpose? Okay, monoclonal antibodies, obviously, they can develop methods to produce large numbers of antibodies required for treatment. They are specific for a particular antigen, and they can be modified so that they do not act as antigens themselves. Interesting. They are specific to particular antigens, and to avoid them being antigens themselves, they could be modified. That is true. So it's 2 and 3 only. C. 14 measles is an infectious disease caused by a virus. The graph shows the number of cases of measles each year in a country before and after a vaccine was introduced. So you have vaccination was introduced, and you could see the number of cases of measles dropped a lot. So what could have caused the decrease in the number of cases of measles after vaccination was introduced? Obviously, when you're vaccinated, that means that you get active immunity. You're injected with an almost less harmful version of the virus, and so obviously this makes the immune system act towards it, and produce memory cells that are going to have the antigens memorized, and so they can produce antibodies directly the next time the secondary immune response. And so obviously the secondary immune response is now faster. We all have secondary immune responses, and so there's no place for the this guy measles to be a host, because everyone here has immunity against it. And so vaccines provided artificial active immunity. That is true. Vaccines provided artificial passive immunity. No, actually that's wrong. It's not passive. It's not just something that will be used once and then it's not a rare occurrence that we're just going to give someone antibodies, and then they can get attacked once and then fine, next time you get attacked you're back to square one. Okay, passive immunity does not really help with the long-term immunity, does not produce memory cells later on, and so it could allow us to act as hosts later on in life. And so no, obviously it's not passive, and fewer people are able to act as hosts for the virus, so yes, it's going to be A. And so this is it for today's video, guys. I hope this video was useful to you. If it was, please make sure to like and subscribe and share this video with anyone who might need it. And so thank you again for watching. Goodbye.